We want to address now the structure of membranes. So we know that every cell must have a plasma membrane, and that is going to be the essential boundary between what we consider the living cytoplasm and the environment. So this defines a cell. We know in eukaryotes there is more than just a plasma membrane. We've in fact partitioned the cell into many compartments, and what's partitioning are more cellular membranes, right? Now the thing about membranes are that they're relatively complex, and they're super important. Membranes are fascinating, and it comes down to this idea of selective permeability. The membranes can't be completely impassable. If they were, you'd have a closed system, and closed systems die. They can't just let anything through either, because then you don't have a barrier at all. So they have to be able to let certain things through when it's appropriate. And this, again, is called selective permeability. Now, in this talk, like I said, let's look at the basic structure of the membrane. And that base layer, our foundation of the membrane, is going to be our amphipathic molecules called phospholipids. Now, we looked at phospholipids when we looked at lipids, so you can go back to that recording and review. But remember, amphipathic means part of the molecule is hydrophobic and part of the molecule is hydrophilic. And you may want to review our hydrophilic and hydrophobic forces to understand this a little bit better. But what we're going to see is that when we put a bunch of those phospholipids together, we can form a self-assembling bilayer. And that bilayer is, in essence, a two-dimensional fluid. So think of a soap bubble. It's incredibly thin. But within that layer of thinness of soap, you can flow. So here we can see that formation of that bilayer with the phospholipids, right? So again, the hydrophilic head groups are going to want to interact with water, and the hydrophobic tail groups are going to try to hide away from water. And again, this will assemble spontaneously. Now, the lipid bilayer, the phospholipids, aren't sufficient to make it selectively permeable. We're going to add proteins to the membrane to do that. Why do we add proteins? Because what do proteins do? They do stuff. And the stuff they're going to do here is help to move things across the membrane. So, we'll talk about membrane proteins specifically a little bit later. But we know that we've embedded these proteins in the membrane. We're creating more complexity in this biological membrane. So again, the fluidity of this membrane, because it's this two-dimensional fluid with the proteins kind of anchored in a sense that they can't move out of the membrane, but they can move laterally within the membrane. Okay, so the lipids are drifting laterally. Rarely do they flip, but then to demonstrate that this, what we call the fluid mosaic model, is a good approximation of what's going on, is you can take two cells. In this case, we have a human cell and a mouse cell. Each have all of the membrane proteins labeled with different colors. So the mouse proteins are purple or pink, and the human proteins are purple. And if you fuse these two cells together, if there was not fluidity to the membrane, what you would see is you'd have a single cell now with one hemisphere being pink labeled proteins and the other hemisphere being purple labeled proteins. But in fact, that's not what you see. You see a mixing of these proteins, a movement of these proteins. Again, this is strong evidence that the lipid bilayer is in fact fluid in a lateral direction. Now, that fluidity can be influenced by temperature. So what we see is that as the temperature cools, membranes tend to get less and less fluid. So think about when you cook bacon or you have fat from meat that you're cooking. In the pan, it's clear liquid. But when you pour it off the pan into a can or into a jar, you leave it in the pan. When it cools, it becomes solid. So in that case, at room temperature, that particular kind of lipid is solid, where at elevated temperatures it's fluid. But we want our living cells to have fluidity in their membranes. Right? So what we're going to see is that by adjusting the types of lipids in the membrane, we can, in fact, adjust the tolerance for what temperature range that membrane stays fluid. All right? So going back again to our talk on lipids, we talked about saturated versus unsaturated. And we know that unsaturated lipids, the fatty acid, has a double double bond between two carbons, and that's going to cause a kink in the chain. Well, when we take those types of fatty acids and incorporate them into our phospholipids and make a membrane out of them, you can see how they tend to keep the packing of the phospholipids much, much more open. So this then contributes to much more fluidity of the membrane because literally there's less interaction, friction, there's less chance for van der Waals forces to cause those adjacent lipids to stay together. So as you can see then with a membrane that's made with simply saturated hydrocarbons, all those straight tails can line up and pack right tightly next to each other, increasing the viscosity of that membrane. So that particular lipid is going to go solid on us at a much higher temperature than one that has unsaturated hydrocarbons. Now, the other type of lipid that we talked about when we talked about lipids as a class of biomacromolecules were steroids. And cholesterol is one such steroid. Remember, it has a double ring structure. And cholesterol is important for membrane fluidity. It's going to act as a buffer for temperature. Now, when you take that mostly hydrophobic, almost completely hydrophobic molecule and jam it into the hydrophobic region of the lipid bilayer, what it's going to do is that it's going to restrain the movement of the phospholipids as they become much more fluid at a higher temperature. But at the same time, because of its blocky nature and its limited ability to kind of interact with the hydrophobic tails, just based on its shape, it's also going to maintain fluidity, prevent them from packing so tightly as the temperature cools. So again, going back to our idea of a very general sense of a buffer, something that lessens the impact. So cholesterol in the membrane is going to lessen the impact of temperature extremes. So again, here you can see an illustration of how cholesterol with that bulky shape is going to pack itself into the membrane. All right.